Hi class, this video will finish up chapter 2 of anaerobic metabolism. So we're going to go ahead and get started with part 4. So part 4 will cover anaerobic adaptations to both the ATP-PC system and glycolysis. And then we'll look at the interactions between aerobic and anaerobic metabolism during exercise. All right, so when we think of the word adaptation, we want to think of what happens to the body with chronic long-term training. So for this first system, we want to talk or mainly think about like high intensity, short duration exercise. When we perform that type of exercise for a very long duration, these are the changes that may take place to the body. So first is an increase in the activity of the major enzymes. The benefit of this, it may result in faster ATP regeneration, resulting in increased performance of short duration, high power activities like resistance training and sprints. One study found there was an increase by about 14% in those major enzymes after isokinetic resistance training. And an increase in 44% in those enzymes after maximal sprints. So what enzymes are we talking about here? Think about the major enzyme that we discussed in the phosphagen system, and that enzyme was creatine kinase. So creatine kinase may increase in the rate that it breaks apart phosphocreatine. And if it can break apart phosphocreatine at a faster rate, we may have faster ATP regeneration, meaning we can get back our energy quicker and we can exercise for a longer period of time before depletion occurs. So yeah, that's our first major change. The second change is elevated resting concentrations of PCR, which is creatine phosphate, or phosphocreatine, and ATP. So I have some results from a study that talked about this number 2 change. So increased resting concentrations means we have more of these substrate or more of these fuel sources even at a rested state. After 5 months of resistance training, one study found resting concentrations increased by about 22% for creatine phosphate, about 18% for ATP, and that resulted in a 28% increase in strength. So benefit of resistance training for 5 months or longer is we have more energy stores. So our body is able to store more ATP and creatine phosphate to help us in this performance, which may also increase our strength. Next, we have sprint type training. So this study looked at 6 weeks of sprint training, and what's interesting is the resting levels remained unchanged. So there was no greater resting concentration of ATP or phosphocreatine in the body, but performance did improve. So what they found was a decrease in 40-second sprint time, meaning we can run a 40-second sprint faster, and improved repeat sprint ability by 2%. They did 6 40-meter sprints with 24 seconds of rest. So that was the protocol they did. And performance did improve without a significant increase in ATP or phosphocreatine. So what this study concluded was that it may depend on if depletion occurred during training. So in order for resting levels to increase above baseline, there usually has to be a complete depletion of the fuel sources. So maybe the sprint time was too short of a duration that the depletion did not occur during training. It's been shown, to deplete by only 20 to 60% of pre-exercise value in a 10 to 30 second sprint. So maybe that 10 to 30 second sprint was not of long enough duration to show a greater depletion. And so that's why the fuel sources remained unchanged, but performance did improve, meaning that sprint performance is not solely based on our ATP and phosphocreatine stores. Last is endurance training. So endurance training shows no significant effect on plasma concentrations, mainly because endurance type exercise does not rely on ATP and creatine phosphate for fuel, and it doesn't deplete the fuel sources. I have a little point down here. More recent studies have actually shown increases. You can see these studies are a little older. This study was in 2002, but more recent studies may show a slight increase in ATP and creatine phosphate resting levels. So elevations in resting concentrations are not necessary to enhance short-term high-intensity performance because other adaptations may be occurring that increases our performance. So one question I get frequently is if phosphocreatine, if that fuel source contains creatine, then what about creatine supplementation for improving high-intensity short-term durations? How does that build up those resting concentrations? And so I have this little video that we'll watch that kind of shows what's the point of creatine supplementation and how does it actually help.
First, a quick refresher. To understand how creatine works, you need to understand why exactly it is your 1 rep max is less than your 10 rep max. One of the major reasons is throughout a set, your muscles may seem to lose strength, but what they are really losing is energy. While we rest between sets, most of this energy comes back to us. Our muscles get their energy from a molecule called ATP, adenosine triphosphate. Triphosphate for the 3 phosphate molecules attached to it. When the 3rd phosphate on the chain gets released, a burst of energy is given off. This is what powers a muscle contraction. Your muscles begin a set, filled to max capacity with ATP. The problem is, ATP is an unstable molecule, so your muscles can only store a limited amount. Let's see what happens when you hit that first rep. Boom, you explode. This is enough stored energy to cover the first few seconds of a sprint, or get you at least a few seconds into that big 1 rep max. 1, 2, 3, now what? We've done 3 seconds and we're out of power already? Well, not so fast, because your body has a trick up its sleeve. When your body uses an ATP, it only uses the 3rd phosphate in the chain, so there should still be something left over, right? There is, adenosine diphosphate. Without that 3rd phosphate though, they are pretty useless, and that's where something incredible happens, because your body has already planned for this. Embedded in your DNA is a gene, an instruction to your cells to produce an enzyme. That enzyme is called creatine kinase. Enzymes exist in cells and facilitate some sort of cellular reaction. Think of an enzyme like a middleman, but what is it connecting? Well, stored in your muscles and ready to be deployed at a moment's notice is another molecule, creatine phosphate. These molecules were sent here by your liver and are holding on to something just for this moment, their own phosphate. And this is their moment to use it. In an instant, they activate, rapidly binding with ADP molecules, giving up their phosphate, reviving those ADPs into fresh ATPs, which can continue powering your muscles. This is why your 4 rep max is still around 90% of your 1 rep max. This creatine phosphate has managed to take an initial supply of ATP and recycle it to last nearly 10 seconds. As an interesting aside, studies have found that baseline creatine kinase levels are higher in African Americans than in whites. So if you're African American, you can enjoy an explosive advantage there. But the real limiting factor isn't the number of creatine kinase connectors. They need something to connect. So the true limiting factor is the creatine phosphate supply. When it runs out, your body is forced to notch down to its next most powerful energy system, the anaerobic system, which, although good, can't produce the same amount of energy as the phosphagen system. This is why your 12 rep max weight is so much less than your 3 rep max weight. So you see, creatine kinase enzymes are found naturally in our cells, along with a supply of creatine phosphate to revive those ADPs into fresh energy. This is our body's own system. It's called the phosphagen system. The issue becomes that your muscles are rarely filled to their creatine phosphate capacity. When you ingest a creatine monohydrate supplement, your body turns it into creatine phosphate and shuttles it into these vacant spaces, allowing your body's energy reviving trick, the phosphagen system, to be used to its full ability. It's no wonder that by the early 90s, Olympic athletes the world over were taking this supplement. Now, the foods you eat do contain some creatine, with the highest amounts in foods such as beef and chicken. But according to a survey, the average 19 to 39-year-old male gets just 1 gram in a day, with women getting even less. This is why if you consume 5 grams of creatine supplement, the recommended amount, it's been shown to be 80 to 100% bioavailable, and translates into a 15 to 20% increase in creatine phosphate stores in your muscles.
So that video continues, and feel free to watch it on your own, but I thought it was just pretty interesting to kind of give you some more information. This infographic here also just talks about, if you're curious about creatine, how does it actually improve performance? So you can also read this on your own. It talks about strength, power, cognition, muscle mass, preventing fatigue, and all of that. But let's move on to training adaptations for glycolysis. The next training adaptation is changes to glycolytic enzymes in the system that we learned about previously. So glycogen phosphorylase, if you remember what this enzyme does, it breaks down intermuscular glycogen into glucose 1-phosphate. This adaptation, when this enzyme will increase or become more efficient, it allows a more rapid breakdown of glycogen for faster energy by increasing ATP availability. The next enzyme that may undergo changes is phosphofructokinase, which we know is the rate-limiting enzyme. So if we have changes to PFK, we may see increased rate of the entire 10 steps of glycolysis, resulting in greater ATP production over time. And the last is lactate dehydrogenase. We know that LDH converts pyruvate to lactate. So this change may not directly influence performance because it's not the rate-limiting enzyme, but it will probably just result in more faster conversion of pyruvate to lactate. So how may these changes to enzymes improve performance? Kind of just went over it, but it's mainly, the main result is faster ATP production, so we can exercise for a longer period of time before fatigue. What type of training promotes these adaptations? So changes have been shown with resistance training, sprint training, and some types of endurance training. The results are mixed, and it depends on the specific features of the training program, such as how intense was the workout, what was the volume of the workout, and the duration of the workout. So basically all types of training may influence changes to glycolytic enzymes. It just depends on the intensity, duration, and the volume. So next is increase in intermuscular glycogen stores. So our body has an improved ability to store more intermuscular glycogen. This will result in less depletion and the ability to generate more ATP for energy. Both glycolytic and aerobic production is affected with increased glycogen stores. So glycogen concentration is greater in the liver by about 10%, but the muscle, which has 2% by weight, but we have more muscle mass. So if we have more muscle mass, we're going to have more intermuscular stores of glycogen than the liver. So we already mentioned that the liver stores, I don't know if we mentioned this actually, the liver stores about 100 grams of glycogen to maintain blood glucose for the entire body, where the muscle has about 400 grams of glycogen. And this meets our glucose demands for the exercising muscle. So what type of training will promote increased glycogen stores? So both aerobic and anaerobic training. Endurance training increases glycogen stores, and this is well accepted by research. So we know that aerobic training uses our glycogen stores and possibly depletes those stores. So we're going to see an increase in our resting or baseline level. Our anaerobic training, like resistance training and sprints, that's shown mixed results. Again, it depends on the length of the program, the intensity, type of training, kind of all of that. But shorter sprints and short burst of resistance training has shown no change if the work is less than 10 seconds. But longer sprints and longer bouts of resistance training, longer than 10 seconds, increases in glycogen production of ATP becomes more important at that point. And so we do see changes to glycogen stores with the longer duration versus the short duration. And then the length of program, it's been shown that 6 to 8 weeks or longer is going to be the best resultant of changes to intermuscular glycogen levels. So here is a muscle, and this is a glycogen granule. So it's about 10 to 40 nanometers, and this is in like the systole of the muscle cell. We'll talk about how the body stores glycogen later, but storage of glycogen and amount of stored glycogen depends on how much muscle mass you have, your diet, so how much glycogen you actually consume or carbs you consume, the type of training that you do, etc. So we'll talk more about that in chapter 3. All right, our third adaptation is something called improved buffering capacity. So what is improved buffering? This is the ability to buffer acidity of hydrogen ions. When you buffer something, it just means to clear away. So the clearing away of hydrogen ions, which reduces acidity in the muscle cells, will improve high-intensity exercise, allowing you to exercise longer before fatigue actually occurs. So the body has these intercellular buffers. So these are our intercellular buffers in our skeletal muscle. And bicarbonate, we see right here, will minimize hydrogen distribution by accepting its protons and converting that acid into something called carbonic acid, which is just a weaker acid over here. We see sodium bicarbonate. So that's kind of just like baking soda, actually. It's going to convert a strong acid to a weaker acid. And so we have bicarbonate, phosphate, and proteins. So these are our buffers, and this is kind of what they do. They turn the strong acid into a weaker acid, and that helps us to clear acidity. So it prevents acidity-associated fatigue, because we already know that acidity is one of the main causes of fatigue. And if we can just convert that strong acid into a weaker acid, we can exercise longer, which means we can produce more ATP, making us better athletes. So we're just have improved performance overall. So here's some facts about buffering systems. So I already mentioned sodium bicarbonate is basically just baking soda. So one study found that the greater dosage and the shorter exercise duration, the more likely the bicarbonate buffering system would enhance performance. So supplement with 5 grams of sodium bicarbonate a day to reduce muscle acidity. But too much, if you kind of go over that dose, it may cause an upset stomach. The results conclude effective at improving short-term high-intensity performance when acidity is present, such as a 400-meter sprint, cycling or time trials, sprints, anything of that nature. Ingestion of sodium bicarbonate may enhance performance of exercise in lasting 1 to 10 minutes. So it can't be of that very short duration. It needs to kind of be in this time frame because this time frame is when acidity occurs. So this time frame is when we have high presence of hydrogen ions. And so if we want to buffer those away, we need to exercise kind of at this duration. And then what about beta-alanine? So actually first, let me go back and tell you a little bit more about sodium bicarbonate. Bicarbonate is not just what we ingest like baking soda. Bicarbonate's produced in the kidneys, and this actually raises our pH and decreases the acidity in the muscles. Other studies have found that untrained actually respond better to supplementation because the trained athletes already have those increased cellular buffers acting to clear away acidity. So the untrained, this protocol actually works better because if you're a trained athlete, ingesting more sodium bicarbonate won't actually improve performance because your body has already undergone those adaptations, and so your body already has buffering systems that are effective at clearing away your acidity. Now let's talk about beta-alanine. So beta-alanine is in a lot of pre-workouts, so a lot of supplements. What's the purpose of this? Well, first, it's a non-essential amino acid. Alanine is an amino acid. It's made by the body already. It contributes to hydrogen ion buffering in our large type 2, so our large strong fibers. It produces something called carnosine, which regulates muscle pH. So here's just kind of an infographic about beta-alanine. It decreases the onset of fatigue. So it kind of it makes you feel like you can exercise for a longer duration because it pushes back that onset of fatigue that you feel. So significantly improvement in working capacity and pushes back that fatigue threshold. We also see increased high-intensity anaerobic performance because beta-alanine plays a role in buffering capacity. So when we do like short-term sprints or bouts of resistance training or cycling time trials, it improves your ability to buffer and clear acidity. And then last is improvement in endurance performance. There's inconsistent results here on how beta-alanine works with endurance, but what has been shown is that it does improve anaerobic threshold, which is just pushing back that fatigue threshold and extending your time to exhaustion, allowing you to exercise for longer. So those are our glycolytic adaptations. It says attendance assignment up here, but normally, since we're online, obviously this is not an attendance assignment, but I want you to think about it. So first think about the type of exercise that you do, and think about which training adaptation do you think would benefit your exercise training performance the most. And this is going to be a different answer for everyone depending on what type of training that you do. So for me, I do a lot of cycling. And so with cycling, it's a lot of high intensity time trials. So where you're cycling as fast as you can for 30 seconds, and then you get a little recovery, and then you go as fast as you can again. So that really high intensity exercise, there's something that's occurring in my body limiting my performance ability. So of these 1, 2, and 3 adaptations to glycolysis, which one do you think would be most important for me doing cycling time trials? It would be number 3 for me, because when I'm doing high intensity cycling for 20 to 30 seconds at a maximal effort, that's a lot of acidity. So that's a lot of ATP hydrolysis, a lot of hydrogen ions that need to be buffered away. So improved buffering is actually probably going to be the most important for that type of exercise. I used to supplement with beta-alanine before I would go cycle. And it really did improve performance because I didn't get fatigued as quickly. I could exercise longer before I hit that breaking point where I had to stop. But think about it for you. If you're a runner and you run long distances, intramuscular glycogen stores would probably be most beneficial for you. And if you lift weights, it could probably be any of these actually. Depending on the weightlifting protocol you do, you could benefit from all 3 of those changes. But yeah, just something kind of neat to think about. All right, here is your chapter 2 quiz review. So for all the quizzes, there's so much information that we talk about, and it's really hard if I just said, okay, study everything in chapter 2. You're kind of like, oh my gosh, like that's a lot of information. So every lecture, I will give you a quiz review to tell you kind of what exactly to focus on for your quiz. So you can take a picture of this because this is not in your student version of your PowerPoints. So take a picture and actually make a study guide. Actually jot down information regarding each of these topics. And then you can use that information while you sit and take your quiz. And that's how you're going to do the best on those quizzes. So yeah, that's for you to study and prepare. So we just have a few more slides to wrap up chapter 2. In this next part, we want to talk about interactions between aerobic and anaerobic metabolism. So normally I would put this picture up here, this chart, and I would have you talk to your neighbor and answer this question. So what is suggested by the figure below regarding anaerobic and aerobic interactions? So just think about this for a second, look at it and try to figure out what is it talking about. So it's basically suggesting that most exercise durations, because that's what we see up here at the top, involve a mix of both aerobic and anaerobic energy system contribution. So there's considerable interaction between each system. With the longer duration, we have that greater aerobic involvement that comes in. So which energy system that's going to be active the most, it's dependent on number 1, intensity. So the intensity of our exercise will be the number 1 determinant in which energy system will be the primary contributor. And duration is the second factor. Duration is the second factor that determines which system will be the primary contributor to our ATP production. So we see over here a 1 to 3 second sprint. It's going to be about 100% anaerobic energy contribution, where you're using ATP and creatine phosphate in this here. But there may still be a tiny bit of aerobic metabolism. It's still active. It's not like dead. It's not a dead system. It's still alive and it's still producing energy, but it's just not really contributing to that performance. And then as the duration gets longer, we see that there's more aerobic contribution. And then the longer it gets, the more aerobic comes in. And then when we are performing 2 hours of exercise, it's mostly 100% aerobic. So yeah, but just keep in mind this chart assumes that each duration of sprint or run is performed with the intent to move as fast as possible in a certain amount of time. So this is needed to estimate percent of each energy system contribution. So here's just another kind of explanation of how these systems interact. We see energy system contribution, we already said this, depends on number 1, intensity, and number 2, duration. So this basically statement has led to the estimate of ATP obtained from aerobic and anaerobic during different types of training. So we want to be able to estimate which system is most active. This is going to be used by coaches, trainers to estimate percent training time in each, kind of using each energy system. So I'll kind of give you some examples here. So we can calculate the active burst, so anaerobic, versus the jogging, walking, and rest breaks, aerobic, to assess the percent contribution from each system. So we want to know how much time should be spent training in various activities. So how much time should we spend training our aerobic versus anaerobic system? And all of that varies by the sport, position, and the type of play. This is used as a guideline concerning energy system dependence on a certain system, or energy dependence on a certain system. So my example is soccer here. So soccer on average uses all three energy systems, the most compared to all other sports. It's been estimated that with soccer there's about 21% walking, 60% slow runs and jogs, and then about 19% sprints and ball possession. So we can take a defensive player. We have Julie Ertz, and these is the women's U.S. national team. So Julie Ertz, she's a defensive player. She runs about 6 to 7 miles per game. And then we have Carly Lloyd, who is our midfielder, who runs about 9.5 miles per game. And then we can take Alex Morgan, who's our forward, who only runs about 4 to 5 miles a game. If we think about football, like receivers and cornerbacks only run about 1.25 miles per game. In an average NFL game, there's only about 11 minutes of full play, which is really crazy to think about. Basketball runs about 2.5 to 3 miles a game, and then tennis also runs about 3 miles. So we can basically watch a sport and break down how much time when are they sprinting and doing really active burst, and then when are they doing more aerobic. And that can lead to how much time should we spend during our practice focusing on certain aspects. So that's pretty cool. Next here we have a 3-second sprint. And we know that we mainly use creatine phosphate and ATP, in which we see are the highest energy contributor during this duration. But what happens to the pie chart as the duration of the sprint increases? So from 2 slides ago when we looked at that little chart there, using that information, what's going to happen if this turns into a 10-second sprint or a 30-second sprint? What's going to happen to the slices of this pie here? So you could probably figure out the answer is that the green and the red parts of the pie are going to decrease, and then the blue and the orange part are actually going to increase. So the longer the sprint, the more we have to rely on aerobic metabolism for energy. So other sports that rely predominantly on ATP and creatine phosphate, think of just like really rapid short duration movements like Olympic weightlifting. So doing Olympic style lifts, swinging a golf club, think of vertical jumping, thinking like shot put, any type of throwing in baseball or football, kicking in soccer. All of that relies mainly on our intermuscular storage of ATP and creatine phosphate. But if we extend that to, like, let's say a 6-second cycling sprint, this would now be about 50% or 25 PC, 25 ATP, 35% glycolysis, and then 50% aerobic. So it just kind of gets bigger and bigger, or sorry, 5% aerobic. But yeah, you probably get the picture at this point, is that the longer duration of exercise, the more aerobic contribution. All right, just a few more statements to just kind of wrap this up, and we already gathered this. Longer the sprint depends more on glycolysis and aerobic. So let's look down here. Here's our distance. So we have a 200-meter sprint, 400, 800, and 1500. So you see that anaerobic is going to be predominant in the shorter duration sprints, but as we move to that 800 and 1500, aerobic then becomes the primary contributor. Sprint performance will also decrease if the recovery periods are not long enough. So our performance will become poorer and poorer, and our work output will become less if we aren't recovered, and that's due to not replenishing our ATP and creatine phosphate stores. And then last, short repeat sprints use a greater percent of aerobic than the first, and that's what's shown in this picture here. So we have a 30-second sprint. The first sprint we did, if you look here, we've got a lot of glycolysis going on, about 50% glycolysis. And then we have ATP creatine phosphate and aerobic at the top. We only had 4 minutes of rest, and then we did our second sprint. Look how much the aerobic metabolism, how that changes. So now the aerobic is the primary contributor, even though the sprint is still 30 seconds. It's because we're doing repeat sprints back to back. We're going to use more aerobic than we did on the first bout. So pretty interesting there. All right, this is our overview slide. We're basically done with this lecture. This just kind of gives you information. So anaerobic provides majority energy during high intensity, short duration, maximal effort work. Short sprints, burst of movements, resistance training. Aerobic will provide majority of the energy, long duration, low intensity, such as a marathon or a distance cycling event. But keep in mind, both aerobic and anaerobic metabolism are present at any given time. Some are just more prominent than others during specific activities. The percent of ATP from each of those sources during various activities can be estimated, but there's individual variation because every human being's body works differently, and everybody's metabolism will be slightly different in which system is being or providing more energy than others during certain activities. But yeah, so that's the end of chapter 2. Now you can complete your assignment. There was one question that we were able to answer in part 4 after kind of going over everything. And then you can also take your chapter 2 quiz and submit that assignment when you're ready. But yeah, so I hope you enjoyed this. This lecture was probably one of the most important because now we can take anaerobic metabolism and continue to talk about it throughout the rest of the semester. So let me know if you have any questions. But other than that, good luck, and I will see you in the next video.